CRISPR-Cas9 is a gene editing tool composed of a CRISPR system as well as a Cas gene. CRISPR-Cas9 systems are highly diverse mechanisms of adaptable immunity used by many bacteria and archaea to protect themselves from invading viruses, plasmids, and other foreign nucleic acids. CRISPR stands for clustered regularly interspaced short palindromic repeats, and Cas9 stands for CRISPR-associated protein 9. To begin, I will go into describing where CRISPR was derived from. According to Harvard University, it is adapted from a naturally occurring genome editing system in bacteria. Here is how it works. A virus invades a bacterial cell. The bacteria captures snippets of DNA from an invading virus and uses them to create DNA segments known as CRISPR arrays. A CRISPR array allows bacteria to remember the invading virus and other closely related viruses. From here, CRISPR RNA is formed after the CRISPR sequence is transcribed and processed. The viral DNA feeds into the Cas9 protein until the CRISPR array aligns with the viral DNA. The CRISPR RNA guides molecular machinery to target and destroy the invading viral genome.
So now that we've talked a little bit about CRISPR and how it was derived, let's go into a little bit more depth about the CRISPR-Cas9 system and how we use it. CRISPR creates a small piece of RNA through a guide sequence that binds to the specific target sequences of DNA and the Cas9 enzyme. Cas9 is then used to cut the DNA at a specific sequence and uses the cell's DNA repair machinery to add or delete pieces of genetic material or to make changes to the DNA with a customized DNA sequence. Furthermore, the Broad Institute explains that research into CRISPR characteristic became known in 1993 when Francisco Mojica reported that sequences at the CRISPR locus matched snippets from the genomes of bacteriophage and led him to correctly hypothesize that CRISPR was an adaptive immune system. Following Francisco Mojica, Alexander Boulton studied bacteria Streptococcus thermophilus and discovered a protein known as Cas9. These two discoveries then led to a domino effect of findings dealing with CRISPR-Cas9, including adaptive immunity, functioning heterology in other species, Cas9-mediated cleavage of target DNA, genome editing, etc.
According to a research article published in Nature in 2018, Gall claimed that CRISPR-Cas9 technology has revolutionized research in agriculture. CRISPR technologies in the agriculture field are a new breeding method that can produce corn, tomatoes, and maize in a more efficient, faster, and cheaper way. Practical applications of CRISPR tools for sustainable agriculture can specifically improve bacteria resistance against stress conditions and therefore increase the productivity of the fermentation process, which ultimately enhances the quality of food products.
Since the nature of CRISPR allows for the removal of unwanted genes, CRISPR plays a vital role in the prevention and treatment of many human diseases, such as cystic fibrosis, hemophilia, and sickle cell disease. In particular, in patients with cystic fibrosis, a gene called cystic fibrosis transmembrane conductance regulator, or CFTR, is known to cause the associated CFTR protein to become dysfunctional. However, with the intervention of CRISPR, this gene has been observed to be repaired by the cells. Additionally, Cas9 has been utilized as an antimicrobial agent and used to target some life-threatening strains of bacteria. Some have also hypothesized about CRISPR's usefulness in helping kill cancer, HIV, and mental illnesses.